in the market. Same way, our second product is the hepatitis B vaccine. All of we know vaccine is just used to prevent some diseases. So here also the development of hepatitis B vaccine is done with the help of by using yeast cells. So what happens is the antigens which is actually responsible to cause that hepatitis B, which is shortly known as HBs antigen, so that is inserted inside the yeast cells, but yeast cells are not pathogenic, so they will not develop any symptoms of hepatitis into the humans. So if we use such type of modified cells which are having genes which will also produce only antigens, not the disease, then it can be used as the vaccine. So for the hepatitis B vaccine, such type of modified yeast cells can be used. This is the photo of such type of commercially available cells which can be used or known as recombinant hepatitis B vaccine. Now second is the another product which is known as the human growth hormone. So generally it is a peptide hormone, and this can be used for the stimulation of growth or to prevent dwarfism into the humans. So such type of product or the human growth hormone, which is shortly known as HGH, then it can be produced generally by the somatotroph cells within the pituitary gland. So this type of materials can be produced by using recombinant microorganisms. So instead of somatotroph, now it is known as the somatotropin, and this type of human growth hormones are known as HGH in humans. Our next important discussion is about the ice minus bacteria, which are generally used for the protection of plants against the frost. So it was first discovered in year 1987, and the strain was Pseudomonas syringae. So it was actually the first genetically modified organism to be released into the environment, because it's not easy to release such organisms which are unknown to us or which were not previously present in the environment. If suppose we release such organisms, then we don't know what might be their effect on the nature. So this was the first experimental microorganism, we can say Pseudomonas syringae, that was released into the nature, and very interesting results were obtained by these organisms. So first it was applied on a plant which we also see after some slides. So a strawberry field in California was sprayed with the ice minus strain of Pseudomonas syringae just before a frost. Means it prevents the frost into the plants, and we can save such type of plants from its harmful effects. So it ultimately increases the crop production, or we can say it saves our economy. Then if we'll see the next product of our interest are the interferons. So interferons are the proteins which made or released by the host cells in response to the presence of pathogens. So particularly they are used against the viruses, or sometimes bacteria or parasites, or even in the treatment of tumor cells. So such type of interferons can be produced with the help of recombinant E. coli. So they are also known as anti-AIDS. So against viruses they can be used. So they are very useful in the treatment of AIDS also, though they are not means the human trials are not yet performed or even given permissions, but they are tested successfully in case of mice. This is some of the commercial brand which is available as interferons, which is known as Interdog, and the various doses of such type of products. Now if we'll see the term bioremediation, as I have discussed earlier, it is the use of microorganisms or their metabolism to remove pollutants. So generally these are the various terms, or bioleaching, or land farming, or bioreactor, composting, bioaugmentation. All these processes are actually one type of environmental cleanup processes. So they will us to keep our environment very safe, and what we can say also they are economically beneficial, as at the end product like composting. So at the end we can produce some of the economically important product like compost. So they will clean up the environment also, and they will give us the benefit also. Some of the examples of such microorganisms which can be used for the cleanup or the removal of toxic materials is the Pseudomonas strain, Pseudomonas putida, or E. coli strain, or the Clostridium testosterone, or some of the E. coli variants which can be produced, or these are the genetically modified microorganisms. And also you can see the various substrates like benzoate or benzene or toluene as in on your screen. Now if we'll see some another important products, then one of the important products is the biopolymers. So means the generally polymers we are using for as a plastic. So and plastic, we all know it creates many environmental problems. So by the help of either plants or microorganism, it can be produced with using renewable resources, means the resources are unlimited, and also they are biodegradable or eco-friendly materials. So generally the strains of Bacillus subtilis or Alcaligens eutrophus or various like corn plant material can also be used. Now what is the exact importance of this biopolymers is not only the economic concept, but also it will affect our environment. Means generally the plastics are not easily degradable, or it will take several years, 100 to 200 years in the soil. But if suppose we are using such type of plastics which are biodegradable, or what we can say, if they are the biological origin, so we call it as biopolymers, then they are easily degradable, and after within few weeks to few months they will just degrade in the soil, and they will not cause any pollution problem. So we can solve the environmental cleanup also, and they are having the same capacity, or what we can say the properties which are very competitive with the commercial plastic which is available, chemically synthesized plastic. Now our next product, if you see, is the recombinant virus. So what is such type? Generally viruses we considered as harmful organism, but sometimes a virus which produces or which recombines the pieces or using recombinant DNA technology, then it can be used to produce vaccines or a gene therapy vectors. Means we are inserting the genes with the help of such viruses, then it can be very positive technology for us. So if we'll see the examples of such type of this, then recombinant vaccinia virus having cloned heme agglutinin gene for influenza virus, so it will protect against influenza virus, though it is a different type of virus, so it will not create any infection, but it will give us the protection. Now you can see such vial which are commercially available. So another example is the genetically engineered fungi. So what is that? So the genes from the Scorpio is inserted in such type of fungi. So how it can be useful? So scorpion toxin encoding genes are used to create a hypervirulent fungus. So fungus itself will produce such type of toxic materials. So it will kill the various insects. So and mosquitoes. So what happens, it will give the protection against malaria like diseases, or also it can give the protection to the humans in several infections, or to the plants itself to the several infections. So such type of materials, means with the help of inserting such genes, fungi can also be modified. Actually the toxin was scorpion origin, but now it is incorporated into the fungi, so it becomes genetically modified, and which can be useful. So if we'll see in the next slide, now we'll come to the another important groups. After microorganisms, we have to focus on genetically modified plants also. So such plant materials are whose DNA is modified using all the techniques which we have discussed earlier. Means what is our aim, why to produce this type of genetically modified plant, is to give some of the extra characteristics which are not naturally observed in plants according to the our wishes. So like the increased rate of resistance towards the pest or disease resistance, or to protect or fight against some environmental conditions, or the production of some of the important nutrients or pharmaceutical agents which are not naturally produced by the plants. So advantages of, if we'll see what might be the advantages, so as you can say, it gives first the basic things, very high yield compared to the natural products which we call as superfoods. Because why this is known as, because they are cheap, also it can be produced in large quantities, and they are also highly nutritious also. So such plants not produce in high amount, but they can tolerate very extreme climates, for example drying condition or freezing. Or even the scientists have developed a type of tomato that grows in salty environment, because if you'll see the osmotic pressure, then some plants naturally occurring tomatoes cannot be produced in salts. So if we can modify their genes, then they can tolerate very high salt concentration. Not only the growth or nutritional value, it also improves the taste and quality or even aroma of that food. So also they can decrease the time of maturing plants, so we can get the very faster cycles of their growth. They are very resistant to diseases, or even increased shelf life, means we can store such plants for a very long period of time. Now if we'll see what such type of techniques, then it is not like that it is always advantageous to us. There might be the some negative sides of the technology also. Some of the disadvantages or risk which might be possible to occur by using this technology is known as, like, first if we'll see the spread of new or more resistant super weeds and super pests, means they will not be even easily removed from the nature. So once they have grown, then it is very difficult, or even super pests. So by using even the pesticides, we cannot remove such pest if they become very resistant species, because there is always a risk factor remains that we have introduced a new type of species of either microorganisms or plants into the nature. Then we don't know whether in the future we will be able to control it or not. Also some of the taste and safety standards are different according to the countries, so it is also question mark, because some country will approve the technology and some will not. So possible creation of new kinds of weapons also, like biochemical weapons we can say, by which are produced with the help of microorganisms, or what we can say the anthrax, which is caused by bacillus anthracis. So such type of modified organisms, and if we are, or even new kind of microorganisms or viral species can be developed which we have never seen, then we cannot control such type of harmful effect. That is one of the disadvantage of these techniques also. Now if we'll see some of the ethical issues also, then it is a question whether we are playing with a god or tampering with the nature by mixing genes among species. It is really a question to be discussed in very detail. Now we can see some of the products, like very interesting, as we have discussed. These are the blue strawberries among the red strawberries, or golden rice, which is having very high content of vitamin A compared to natural rice, or flavor sour tomato, which is more resistant, having increased shelf life. Or this is a corn which shows us the different colors, means it indicates we can produce corn with various types of medical drugs or amino acids or vitamin sources. Or these are the genetically modified wheat, which is having very increased protein value, or wheat with antibiotics. Or all of we are well aware of the BT cotton, so which can be produced or having a very high quality. And this is the plant which shows luminance with the help of luciferase enzyme. Now our last group of discussion mainly is the GM animals, means it is also the introduction of genes into the germ cells of animals, and we can modify such type of cells. So transgenic animals can be, or the experimental models generally are produced for the research purposes mainly. So various vital discoveries we can develop. So such type of proteins, if we'll see, then stable expressions of human proteins have been developed in many animals including sheep, pigs and rats. So you can all of you aware with the production of a dolly, which is a clone, and these are some of the rats which are produced genetically and showing luminance. Now another very high size, increased size of animal like cow, which is having a very high biomass, so it gives us the meat production compared to the natural cow size. It is a jumbo size cow. So all these are due to the genetical modifications in the animals. Next, gene therapy. Actually, gene therapy is the insertion or alteration or removal of genes from the individual cells or tissues. So it is used to treat the humans from the genetical disorder. So in one sense, actually we are altering something of the human genes. So whether these genes are changed, so we can say it is a genetically modified humans. Not exactly we are modifying the genes, but for the disease treatment purposes we are modifying some part of the cells or genes. So that's why we can even say this is also the genetically modified humans, because earlier they were sensitive to disease, now they are resistant to diseases. Such examples which can be treated with gene therapy are the severe combined immunodeficiency or CGD or hemophilia or blindness or muscular dystrophy. These are the list which can be treated. Now what might be the future prospects of our technology? So this technique can be help in the treatment of cancer, or we can say the various type of material like the fats which we know the exact composition. So it is not that we are randomly taking any fats or harmful effects of that fats, like some of the vaccine as we have discussed earlier, or golden rice as we have seen earlier, which are vitamin A rich, or various transgenic if we'll see the concepts for the genetic improvement of livestock or agricultural are being integration of this technology into practical farming system remains some distance in the future. Means it is not that much easy as we are discussing on paper. So it is, if we'll see the future of these techniques is very bright as far as the various pharmaceutical or anti-cancer or edible materials are concerned.